The four types of viruses pathogenic to humans are negative sense RNA virus, positive sense RNA virus, retrovirus and finally DNA virus. So let's look at a few examples and also I will explain each type. Negative sense RNA viruses include such entities as the common flu, bird flu and probably also the Spanish flu. And what separates this type of virus from its counterpart, the positive sense RNA virus, is the type of RNA that it carries. But let's start by explaining what RNA is. If DNA is the hard disk, then RNA is the RAM and the ribosomes could then be considered the processor. And the difference in the types of RNA vira is the type of RNA they carry. As negative sense carries tRNA while positive sense carries mRNA. But what is tRNA and what do you mean carry? Well, the term carry refers to the fact that all viruses are basically like a wrap, and inside this protein wrap sits the viral RNA and the viral proteins if any. This protein wrapper is then designed to only open under a certain condition. For an example, let's say the fluid's wrapper protein is designed to only open under acidic conditions, and where the virus finds these acidic conditions is then on the sialic acid receptor. But anyways, back to tRNA. What is tRNA? The T stands for transfer. And it needs an enzyme that is called RNA polymerase II, in order to be transcribed to mRNA. But anyways, what is tRNA? Other properties. Yes, for one, it transports the genetic instructions of the DNA out of the cellular nucleus, and secondly, it also is the RNA version of DNA, i.e. it won't carry amino acids to the ribosomes. And obviously the negative sense RNA viruses must carry their own RNA polymerase II in order to infect a cell. But wait, what are ribosomes? They are the protein factories, i.e. they force the amino acids that the RNA carries to react with one another at the active site of the ribosome of the cell and are made up of RNA and protein themselves. Positive sense RNA viruses. Examples include the crown-shaped virus, SARS, and probably the bubonic plague. These viruses are made up of mRNA, otherwise called messenger RNA. And this RNA is the most stable form of RNA in existence. And it can carry amino acids by hydrogen bonding with them to the ribosomes of the cell. But what is a hydrogen bond? Well, a hydrogen bond is an electric interaction, meaning that it comes from charge, and is a result of the positive charge of a hydrogen atom, and the negative charge of a lone pair on an electronegative atom. Did I fail to mention that the hydrogen must be bonded to an electronegative atom itself in order for a classic hydrogen bond to form? Oh well, but that was a sidetrack. But because the viruses don't have to carry a RNA polymerase II of their own, they can have RNA encoding immune system evasion, a protein that allows them to wreak extra havoc in an infected cell. For an example, they could theoretically carry a protein that disrupts the most important function of a cell, oxygen metabolism. As RNA, of course. Anyways, back to the viruses. How do they form multiple copies of themselves? Well, this they do by copying their mRNA with an enzyme, and then this mRNA is then gobbled up by the capsule protein. And the enzymes the virus needs is a RNA polymerase to copy the viral genome. Negative sense RNA virus needs the same, by the way, and it needs a protease enzyme, an enzyme which cuts proteins to exit the cell. This is by no means the complete list of viral elements, but just what enzymes you need to have a basic positive sense RNA virus.
Sense RNA viruses all until they inject their payload into your cells. From here they activate an enzyme called reverse transcriptase, which translates their RNA genome to DNA. And next up they use an enzyme called integrase, which will put their genome into your genome. Warning: speculation coming up. But is it possible that there exists clean promoter regions in the DNA? What is a promoter region? A promoter region is a unique sequence of about 100 to 1000 BP base pairs which binds something known as a transcription factor, which in turn has an attractive electric field towards RNA polymerase, the enzyme that does the exact opposite of the reverse transcriptase. And let me just add right away that the virus likes to sit still in your genes for years. I explain the virus's genes isn't transcribed/activated for years. And now you might be thinking, a virus which transcribes backwards, it must be possible to use medicinally. And you would be absolutely be correct. Ever heard about something called gene therapy? Prominent examples include Zolgan SMA for the dreaded disease SMA, which fun fact is also the most expensive drug in the world. And cancer treatment with immunotherapy. So how do these things work? Well, allow me to explain. Zolgensma is based on a virus adeno I think which has been modified so that it only targets the motor neurons of the brainstem of the spinal cord. Next up, the virus will inject a correct version of the gene that encodes the faulty protein which is the cause of that disease, which is called survival motor neuron 1, and thus increase the production of that protein. Immunotherapy works by having the virus carry an oncogene, a gene specific to cancerous cells, to the T cells of the immune defense system, which in turn causes the immune defense cells to target the cancer since they do now know how it looks. And examples of these viruses include HIV, AIDS and adenovirus. And finally we have the nanoviruses. Just kidding, but maybe I will do a future video on those, so hit the subscribe button. But instead this is about DNA viruses, which includes HSV, herpes simplex virus, as the only one I can name. And these viruses differs only from the positive sense RNA viruses in the sense of what type of genome they carry. Since they are called DNA viruses, they must be carrying their genome as DNA, right? Ding ding ding, we have a winner. That is absolutely correct. And they are dependent on the integrase enzyme just as the retroviruses. And they are having a similar time frame of evolution as an infection as the retroviruses. Thus meaning that an infection with a DNA virus progresses ultra slowly. This is due to that with RNA viruses it takes literally minutes from a cell is infected to the cell starts producing the viral particles, and this is due to the fact that no transcription is needed in order for the virus to communicate with the ribosome. However, with DNA viruses it can take literally years, and this is due to the fact that first the transcription factor must find the gene. And next the gene must be transcribed. And finally the gene must be transcribed again from tRNA to mRNA. And the rate-limiting step in this process is of course the transcription factor finding the gene and the wait from a transcription factor having found the gene to a RNA polymerase binds the gene. So that's it. Please like, share and subscribe. And also make sure to grab my free e-guide 7 tips to supercharge your immune defense system. Link is in the description.